So I'm going to talk about new treatments for Fragile X syndrome. This has been a focus of my research for many years. Of course, there's the usual treatments for anxiety like SSRIs or stimulants for ADHD, but I'm going to talk about targeted treatments that help to reverse the neurobiological abnormalities in Fragile X, because we're very interested in reversing the intellectual disabilities that these individuals have. So there are many advances in treatment. First of all, it's important to know that Fragile X is the most common inherited cause of intellectual disability. In most countries, it represents anywhere from 5 to, in some countries, up to 10% of those that have intellectual disability. And for those with autism, it represents about 2 to 5%. So anybody that's diagnosed with an intellectual disability or autism needs a Fragile X DNA test for 2 reasons. There are now new treatments that are very exciting that I'm going to talk to you about. And also, Fragile X is a disorder that runs through the family. So the mother of children with Fragile X syndrome are the carriers. And I'm going to talk about those who are carriers with the pre-mutation versus the full mutation in just a minute. And most of the time they have hyperactivity. As you can see, this young man down here has hyperactivity, but about 60% of boys with Fragile X syndrome meet diagnostic criteria for autism, and about 20% of girls with Fragile X syndrome meet the diagnostic criteria DSM-5 for autism. Now, there are 2 types of mutations in Fragile X. The full mutation has a CGG repeat on the front end of the gene, and by CGG, it's the nucleotide repeats on one side of the DNA. DNA is like a twisted ladder. So there's a repetitive CGG repeat on one side of the DNA, and when that repeat goes above 200, the gene turns off and does not produce messenger RNA, which is the copy of the DNA that goes out to the cytoplasm of the cell to produce the Fragile X protein. So in girls who are definitely superior because they have 2 X chromosomes, okay, the girls with the full mutation are producing a limited amount of the Fragile X protein, FMRP, so they're less affected than boys. Now, everybody has this gene. As you can see from the far left, in typical individuals, it's anywhere from 5 to about 40 CGG repeats. The average is 30. This protein is very important for early development. You need this protein to help to mature your brain, and you also need this protein in aging. And those who are carriers or have a smaller CGG expansion that is 55 to 200, these carriers are very common in the general population. About 1 in 200 women is a carrier of the pre-mutation for Fragile X, and about 1 in 400 males is a carrier. Now, the carriers, we know, produce just the opposite of Fragile X syndrome. They produce too much messenger RNA, and this is toxic to the cell because all this excess message sequesters proteins that are important for neuronal survival and the survival of other cells in the body. And so these individuals can have a variety of health problems. In childhood, they can have anxiety and ADHD, and we call that the Fragile X associated neuropsychiatric disorder. And in adults, that presents as anxiety and depression, insomnia, chronic fatigue, and chronic pain. So many of the mothers that we see suffer from these problems, and it comes from RNA toxicity. Now, the most severe problem is the Fragile X-associated tremor ataxia syndrome, or FAXTAS. This we discovered and published in 2001. And now we see hundreds of grandfathers and grandmothers of individuals with Fragile X syndrome who have a tremor and a balance problem, and it's usually misdiagnosed as Parkinson's disease or essential tremor, but in 50% of the males, it can cause dementia. So we're doing a lot of research on these problems. But the premutation also causes early ovarian failure in women. That means menopause before age 40. And that occurs in about 20 to 25% of women with the premutation. So OBGYN doctors are often diagnosing the premutation in women that present with infertility or ovarian problems. Now, I don't have time to talk more about these premutation problems, except for the workshop this afternoon, where I have a family that has experienced all these problems, including Fragile X syndrome, FAXTAS, FAXPOI, and FAXAND, along with some international doctors from our international training program for neurodevelopmental disorders. I have 3 doctors, one from Italy, one from Turkey, and another from Thailand. So come to our workshop, the second workshop this afternoon, and hear about all these other problems. But now I'm going to talk about Fragile X syndrome. And again, remember, Fragile X DNA testing is easy to do, it's covered by insurance, and doctors need to do it when they have a child with intellectual disability or autism, because you need to know the cause so that they can take advantage of some of these new targeted treatments and do genetic counseling for the whole family tree. I saw a family in Indonesia that has 50 members in the family with Fragile X syndrome and intellectual disability and autism. So these families can be quite large, particularly in foreign locations where there's a lot of children born to each family. Now, there are behavioral differences in Fragile X syndrome. They can have anxiety, hyperarousal to sensory stimuli, and sometimes that can lead to aggression if there's too much stimuli. ASD, or autism, as we mentioned, in 60%. Aggression sometimes occurs, poor eye contact, hand flapping, hand biting, a variety of stereotypic behaviors, and that's what gives them often a diagnosis of autism. Here are 3 brothers, all with the full mutation from one family. So hand stereotypies, poor eye contact, and hand biting are features that are very common in Fragile X syndrome. Now, in order to know about targeted treatments, we have to talk about a few molecular things. Now, I know that's hard to think about molecular pathways, but I made it simple. When the Fragile X protein is missing, that screws up many pathways that get dysregulated. Because the Fragile X protein, or FMRP, is the regulator for translation, that means translating messenger RNA into their respective protein for at least 30 to 40% of all the genes that cause autism. So when you're missing the Fragile X protein, a lot of pathways get dysregulated. And the targeted treatments are really focused on 3 different pathways that I'm going to talk about. The mTOR pathway, which you see that M and then TOR pathway, this is downregulated by metformin, a very common treatment that I'll mention, and we're seeing some great results. The CBD pathways, and we're using topical cannabidiol, or CBD, to help with behavior problems in Fragile X, and also cyclic AMP, which is the energy compound that allows neurons to connect. Cyclic AMP is too low, so there's a new treatment called zatomalast that inhibits the enzyme that breaks down cyclic AMP. So guess what? Cyclic AMP goes up, and you are having more synaptic connections, And guess what? That improves intellectual disability and cognitive abilities in individuals, particularly males who have been studied with Fragile X syndrome. So I'm going to show you the studies, and then we'll end up with a short discussion about gene therapy, which is coming, and it is quite exciting. Now, one medication that I've used very early on in the treatment of Fragile X is sertraline, which is also called Zoloft. And at a synapse, the SSRIs, or selective serotonin reuptake inhibitors, allow more serotonin to stay in the synapse. And there is evidence that those with autism have low serotonin at the synapse, which is one of the reasons why I started using serotonin at low doses early on in childhood. And serotonin and sometimes other SSRIs like Prozac can lower anxiety, improve language, and help with social interactions. And so we did a controlled trial of very low dose sertraline, just 2.5 to 5 milligrams in young children with Fragile X, ages 2 to 6, and we found significant differences compared to placebo. We found improvement in visual reception, motor abilities. There's the p-value there. These are all from the Mullen scales of infant development, and it shows how much better or how many months better on sertraline versus placebo on the lower end there. And it did help with the language, but not significantly so, although there was a significant improvement in those that had autism and Fragile X syndrome. And so I'm going to move on to topical CBD ointment, which is the Zynerba study, and putting this ointment on the arms twice a day has been very helpful for those with Fragile X, and I think CBD will help many other subtypes of autism and other disorders. And in the control trial, for those, if you look at the bottom part of that scale, that had greater than 90% methylation, meaning that their full mutation was turned off in the majority of cells, it was significantly improved on CBD, which is the purple, compared to placebo, particularly for anxiety and social interaction and behavior problems. Now I'm going to just mention about metformin, which is a type 2 diabetes medication. But metformin has a variety of other very positive features. The mTOR pathway that I showed you is too active in Fragile X, and so metformin brings it down to normal. It was first tried in animal models of Fragile X syndrome, and then IQ can decrease, and also individuals with Fragile X can overeat, and they have too much weight gain, and also their testicles are too big. We call that macroorchidism. So the testicles are about 2 to 3 times normal in Fragile X. And when we treated them with a controlled trial of metformin that happened at 2 sites in Canada besides the Mind Institute, because it was funded by the Azraeli Foundation, which is a Canadian foundation, we found that for those ages 6 to 11, there were significant improvements, not only in the primary outcome measure, expressive language sampling, but also in all of the subtests, and the composite score was significantly improved compared to placebo. So these were pretty dramatic effects in those with Fragile X syndrome compared to placebo. However, it didn't work as well in adults. There was improvement in behavior, but not improvement in the language abilities. And so it seems to impact a couple of pathways much more strongly for young individuals with Fragile X syndrome compared to older ones. And those who are mosaic, that means they have some cells with the full mutation and some cells with the pre-mutation, they did great also with metformin. As you can see, the red is metformin, and the scores on the ABC composite score compared to placebo, and also on the Adams scale, significantly improved. So metformin is cheap easy to obtain, it has many positive effects, and it is a new targeted treatment for Fragile X. Now, I just have 1 or 2 minutes left. I want to talk about Zatomalast, and David Hessel on the far left there is one of the psychologists of the mind, and he developed the NIH Toolbox, which is a way to measure cognition via a computer after a short period of time, because you can't repeat the IQ test but once a year. So if we want to look at cognitive improvement, we go to the NIH Toolbox that has been geared for use in those with intellectual disabilities, and the controlled trial of Zatomalast that inhibits the enzyme that breaks down cyclic AMP, so the cyclic AMP levels go up. And they had improvement in visual vocabulary, in language, in reading. And so there's the structure of Zatomalast, and in the top 3 ones there, it was significantly improved, and also by parent scale. It's occurring in many sites throughout the US. So the one study that showed improvement in adults has been published now, and then many sites are working on children 9 and older into adulthood, so we'll look at efficacy very soon for these younger individuals. I'm going to end with a slide about gene therapy. This is actually a slide about how to replace the FMR1 protein. The problem is FMR1 protein doesn't go through the blood-brain barrier, but when you put a TAT tail on the end, it does go through the blood-brain barrier, and you can give this IV, and in animals it reverses Fragile X syndrome, and we hope within the next 2 years we'll be able to do an injection of FMRP that will replace the Fragile X protein that's missing in Fragile X syndrome. The one downside is you have to do it weekly, but we hope to be able to start that and really dramatically reverse this disorder. I'm going to talk about treatment of pre-mutation problems this afternoon, so I hope to see some of you back this afternoon for our workshop. And thank you very much. It has been a pleasure, and I have a great team.
I wanted to start out with just some talk about my perspective and some of the language I'll be using. It's important just to point out that my perspective has been shaped by my experiences as a parent of 2 children with neurodevelopmental disorders, and also that our research is guided and informed by the families that participate in our research studies, and you'll hear an example of that today. And I tend to use person-first language because that's what most of our families in our studies tend to prefer. So I wanted to start out just by talking about kind of an overview of the kind of research I do. I've been studying brain development since 1990s, and my goal is really to understand how early life events and things that could potentially be modified could lead to later changes in neurodevelopment and also resulting neurodevelopmental conditions. I've been primarily focused on autism spectrum disorders and more recently ADHD, attention deficit hyperactivity disorder. And also as an epidemiologist and a molecular epidemiologist, I'm really focused on learning more about some of the biologic mechanisms that could be playing roles in between the path to leading to some of these different trajectories of development. And today I'm going to focus on one particular exposure that could happen early in life. And I'm going to talk about, because it's fairly relevant as we start to go into the wildfire risk period. So I'm going to talk a little bit about wildfire smoke. Most of my work has also come out of the MARBLE studies, as has been mentioned. That's the Markers of Autism Risk in Babies, Learning Early Signs. That's what the full MARBLE's acronym stands for. As mentioned, this was a study started way back in 2006, where we enroll families that have a child with autism already, and then the mom is pregnant with a new child, and so we follow that pregnancy and that child until they're older, and we assess them for various neurodevelopmental outcomes, including autism. And we do that in the Mind Institute Clinic using gold standard methods. And these younger siblings of children with autism are at much higher risk for developing autism themselves. It's about 7 times the risk of the general population at about 1 in 5 or 20%, compared to 1 in 31, the going rate in the general population. And so you might remember back in 2017, in the Napa, Sonoma, we had large wildfires that burned neighborhoods. And so this was one of the most devastating wildfires at that time. And then it was followed in 2018 by the Camp Fire, which then became one of the most devastating fires in Northern California history. So several of the largest and most destructive fires have happened in the last several years, and these have produced some of the highest particulate matter concentrations ever recorded. But the other concerning factor is that they're hitting these urban areas, which is not only a concern for lives lost and property loss, but also in terms of what's going into the smoke. Because when these wildfires hit these areas, now they're burning things like cars and melting refrigerators and burning building materials and things that release other chemicals into the smoke that might be of concern and travel far distances in the air. So when these wildfires broke out, these women in our Marble study who were pregnant were asking us at the time how concerned should they be about this wildfire smoke and how they should protect themselves and their developing baby. And at the time, we found out we didn't really know much about the impacts of wildfire smoke on pregnancy and children. And so we did know that during the period of early development, there's a lot of changes that are happening, and this is a period that's particularly vulnerable to various environmental exposures, including air pollution in general, like ambient air pollution from traffic and industry and other things. And there have been lots of studies linking air pollution in general during pregnancy to other birth outcomes like birth weight and gestational age or preterm birth. And there's also studies showing links between air pollution, high air pollution levels, and increased risk for other neurodevelopmental outcomes like autism and ADHD. But we didn't really know this for wildfire smoke particularly, and the components are a bit different, although some of them overlap. And so we decided, this might be worth investing because there's a lot of reason to think that there could be some sort of effect of having the exposure to wildfire smoke during pregnancy on that developing child. We already knew that smoke exposure could lead to black carbon from the smoke getting to the fetal side of the placenta, so it's getting in there. And we also know that components that are found in wildfire smoke, like particulate matter, can trigger inflammatory damage, which can also influence placenta function. And so these were things already known from air pollution studies, so we would think that maybe wildfire would have similar effects. And then we also saw some studies came out from earlier wildfires showing that there were links between wildfire exposure in pregnancy and preterm birth and low birth weight. And so there were still questions, though, on who's most vulnerable, and whether those early developmental impacts could lead to longer-term impacts on child health outcomes, and also what mechanisms are going on in order to be able to kind of maybe reduce some of those effects. And so I've been very interested in just, how do these early life exposures make a difference for years later, the neurodevelopmental outcomes of some of these children. And this kind of leads me to the theory that's been around for a while now, showing that there are these developmental origins of health and disease, and adult disease at that. So we know that there's this time of kind of reprogramming that goes on using epigenetics. So epigenetics is, a lot of time we focus on DNA methylation, which is this little methyl group that's put on DNA, which helps, that's associated with gene expression. And so the important part of that isn't really that mechanism, but during early development, around the time of conception, so very early on, the whole methylome tends to be wiped out for that developing fetus and then reestablished, except for at certain imprinted genes that doesn't happen. And we think the reason for this is to kind of adapt that developing fetus for its outcoming environment. And so you can imagine that this is also a period that's really important for maybe environmental influences to make a big mark on how the resulting trajectory of that development and the gene expression that happens after that point. And so we did see evidence of some of this in macaque studies. So, when these wildfires happen, we're not the only ones exposed to the smoke. There's these macaque non-human primates that are sheltered outside, so they just live outdoors, and they're in the smoke more than we are, so keep that in mind, that they're exposed basically all day long. But there were monkeys that were pregnant, and then also ones that had young infants that were exposed to the smoke, and they saw in those infants that they later had influences on their respiratory health and their immune dysfunction, but also that there were changes in their methylome, or the epigenetic markers, that showed changes in their immune and nervous system genes in those pathways. And so that was actually when one of those researchers came to me and said, she was a respiratory researcher, and she's like, it looks like there's something neurodevelopmental going on here. So we've collaborated now to kind of look more into this. But also those infants, as they followed them up, they developed a lot of behavioral and memory impairments. And so this was also a concern that maybe there's these longer term influences of that early life wildfire smoke exposure. And so with all of that, I started a new cohort. This was called the BSAFE, Biospecimen Assessment of Fire Effects, Wildfire Pregnancy Study. So these were all moms that had been pregnant during the time that these wildfires broke out. So starting with the 2017 Napa Sonoma fires, continuing through the 2018 Camp Fire, the 2019 Kincaid Fire, which was much smaller, the 2020 Complex Fires, and then 2021, we also had the Lightning Fire. So there were plenty of fires to follow up on, and over time we enrolled about 550 women into the study, and then we followed their pregnancy outcomes and collected a lot of biological samples close to the time after the wildfires had occurred. And so in these moms, this is the areas where we pulled them from, mostly in the greater Sacramento region, where we could drive up to 2 hours, because we drove to them to collect samples and information. And then the idea is now we're going to be hopefully bringing them all into the mind to assess their children. So this is just showing actually where they had lived at the time the fires broke out. And just a note that some of them evacuated during the fires or moved to other places to avoid the smoke. And those are shown here, but basically most of them stayed in Northern California. We collaborated with Sean Raffus and Mike Kleeman here at UC Davis, did some very fancy air pollution modeling for the wildfire smoke during each of these fires. And then this is just kind of giving you an idea of what the wildfire peaks look like across fires. So you can tell that November 2018 Camp Fire was, again, like much larger than the 2017 fires in terms of concentrations of smoke and particulate matter that went into the air. It also covered all of Northern California. And then the complex fires in 2020 and 2021 look relatively small, but they lasted a lot longer. And down below you can see what the actual fires look like. Above was what our participants experienced based on their address histories. And so we just recently did a pilot study where we went back to the 2017 and 2018 wildfire families, and we had the mothers complete questionnaires about their children and their behavior and their respiratory health. And the part I'll talk about now is just, these kids were around 4 years old at the time that the moms completed these questionnaires. She did a child behavior checklist, and we did about half males, half females, about 50 of each. And then we connected that with the particulate matter, PM2.5, that the mom would have been exposed to during pregnancy, during the wildfire. And even though all of these moms were exposed to some level of smoke during the fires, there was a pretty good variant of more and less exposure. And what we saw in our findings was pretty concerning. We found that almost across all of the different scales of developmental problems on the child behavior checklist, that those who were exposed to higher PM2.5 or smoke levels had reported more problems on basically all of these different problem scales, including the one for autism and ADHD, internalizing and externalizing. So that was a big concern. And the other thing that was kind of interesting was that this was completely driven by the males. You can see that in the females shown there, when we split them apart, they really didn't show the same level of problems associated with the smoke. It was really driven by the males. The other thing that we noted was that we had only looked at those 2 wildfire years, and if we looked by year, which I'm not showing, but the 2018 Camp Fire was really driving all of those associations as well, which was a much bigger year in terms of much higher concentrations of smoke. And so now we just got funded to bring all of these families in, or at least connect with them again. We hope to get more assessments on everybody. We also want to assess the females now that they're older, because we think that maybe there was some of the problems tend to show up later in girls, and so we might be missing something that might show up later. So now we're doing that work. We're also looking at a lot of our measures to look at kind of mechanisms, like whether there's inflammation and immune responses involved in maternal samples that were collected early on, and then also looking at epigenetic markers in the kids. And I did want to follow up with just that study is ongoing. It's really unique because we have a cohort with samples where we can look at kind of the mechanisms and also exposure levels and things like that. But on the flip side, that was the first study ever looking at this, so we wanted to kind of follow that up. So I have a study that's a statewide linkage study, so much larger. Now we're talking all births in California across 2 decades. And we looked at their levels of wildfire smoke across all of that time, each family, each birth, and what they would have been exposed to during pregnancy. And we have done this, I'm presenting results that are a little early, preliminary, and just from 2010 through 2019. And you can see kind of the areas that were most affected by wildfire smoke there. And just to note that from this study, we also linked all those birth records and their air pollution at that time to later developmental conditions in the Department of Developmental Services data. And we saw that when we looked at autism spectrum disorders in particular, that this is again over 8.6 million records we're looking at, with over 70,000 cases of autism. And we see a similar association between higher wildfire smoke or particulate matter concentrations in the mom during pregnancy was linked to higher risk for the child later being diagnosed and treated for autism spectrum disorders. We did note that there was a threshold effect, so it was only if those concentrations got above a certain level. And then we also saw that these associations were more pronounced for families where they weren't previously exposed to air pollution at high levels. And so it's kind of the new introduction of this environmental exposure that seems to make a bigger impact. So again, we're really interested in looking at mechanisms which might be driving some of these things so we can learn how to mitigate some of these associations. And I've been really focused on placentas because they're a fascinating tissue that at birth we can collect, and it kind of serves as like a time capsule of what happened gestational events that happen during pregnancy. And so we can use that to look at epigenetic markers that show gene pathways that might be influenced not only by the exposure, but then also that relate to later developmental outcomes. And I know this is a lot, but the takeaway is we looked at about 230 placentas, and this is now combining the MARBLES cohort study of high-risk autism families with the Be Safe pregnancy cohort, or where they're exposed to wildfires, kind of putting them all the placentas together, looking at pathways that overlap in terms of changes in methylation during pregnancy, that in relation to both the wildfire smoke exposure and later autism and other child behavior checklist problem behaviors in the child later. And we saw overlap in a couple different modules. And some of these implicated inflammatory response pathways. And interestingly, one of the strongest ones showed the biggest overlap was really implicating synaptic pathways. So you can see like neuronal specification and synapse organization, a lot of these terms for pathways that were both linked to both the exposure and the outcome. So this is work that we plan to follow up. I won't go into the national echo study, but just to let you know that there's also a national study of pregnancy. We have a site here. There's wildfire events that are happening during those pregnancies, and so we're also collecting samples and following those children up for many different outcomes beyond neurodevelopment, including obesity and other respiratory outcomes. I just want to mention there's also other ways we're looking into some of the mechanisms. We're also looking more into those macaque monkeys to look at their epigenetics, and then also exploring brain organoids, because we have collected wildfire smoke samples from all these wildfires, and so we can expose them to brain organoids and see what pathways are influenced. And then also, we use those samples for a lot of other things, looking at which exposures in that wildfire smoke in particular might be of concern. So for example, moms with the 2017 fires were really worried about heavy metals exposure. And so we collected hair and fingernail samples where we can kind of look at a timeline, especially in hair, before and during and after the wildfires. And you can kind of see a little peak there that happens to be tin. But we can kind of time it back and see what exposures that they were exposed to during that time of the fire, and then see if those are later implicated in some of these developmental outcomes. We're also working a lot with trying to get funding to do some intervention studies to put air purifiers in pregnant people's homes, and then also community education about using masks if they're outdoors and that kind of thing. So a lot of different directions. Also, a lot of collaborators to recognize. I just want to recognize Amanda Goodrich, Carl Osharkey, and Logan Williams. Those are all people in our lab that have work that I have presented today, and then also a lot of funding sources and a great team. Some of my disclosures, and thank you for your attention.
Okay, so these are my financial disclosures, mainly NIH, recently Myers Incorporated, and none of their funding influences anything I'll talk about today. So on the left side of the screen, what you'll see is a traditional diagnostic and statistical manual symptoms of ADHD. So not everybody has all these symptoms. Typically is divided into the inattentive type or the hyperactive impulsive, or what we call the combined type. And the inattentive type is much more common as you mature, whereas the hyperactive impulsive is really relevant to preschoolers, and combined type is really common from, I'd say, school age through at least adolescence and sometimes earlier adulthood, but we definitely see it in adulthood as well. What you see on the right side are some of the behavioral outcomes. Now, not everybody is going to have all those outcomes. That's just some of the common outcomes. I can tell you the number one reason why we see people in our clinics is because of educational problems. So their children are having difficulty with their schoolwork. If they're college students, it's the same situation. But we also see we see a lot of stigmatization about ADHD. It's been really interesting over the years how it's really changed in the community. It's it's just changed because I think of social media in so many different ways, and I'm not going to really go into that whole aspect of it at this point, but it definitely has changed. Also, we're finding irritability is particularly common in girls, not as much in boys, but especially the teenage girls. We're finding that in our research. And unfortunately, there seems to be a higher sensitivity and vulnerability for substance use. Again, none of these are true of everybody. And accident rates are much higher. So that's automobile accidents as well as household accidents. Now, I did forget to say, also in terms of the language I'm using language that's person-centered as well as identity-centered, depending upon which study I'm talking about. So forgive me if I misspeak at some point, but we're trying to be as inclusive as possible, and what I've typically done in the past is ask the community how they want to be represented. And I'm also coming from the perspective of particularly the ADHD. I have family members with ADHD. I have lots of colleagues with ADHD. I have trainees with ADHD, but I'm also an active clinician, and almost every research project that I've undertaken has really been influenced by some of my personal observations, particularly in some of our quasi-laboratory studies, like some of our classroom studies, but also from my clinic and parents telling me their stories and adults telling me about their challenges. And that's made me think about what paradigms we use in the context. So this study probably is not much of a surprise to anybody. This is from the Center for Disease Control. We've seen a huge increase in diagnosis in children, but it's leveled off in about 2022, 2023, but over 1 million children more were diagnosed between 2016 and 2022. So quite an increase. But the largest increase is really in adults, and particularly in adult women. That's where we really see this dramatic increase. And a lot of this occurred around the COVID time. And at that point, a lot of adults will tell you they had a very hard time getting their prescriptions filled. I think that has eased a little bit, and I think probably the companies who manufacture the medication were able to increase the amount. But I think really since COVID and TikTok, we're hearing a lot more about ADHD. Now, in terms of the treatment landscape, it really varies, but we can say overall about 1/3 of kids with ADHD are not getting adequate treatment. Treatment typically involves behavioral treatment, parent training. There's some really awesome school interventions as well, and medication, but about at least 1/3 of kids are not getting the combination that we really expect to bring the best outcomes. So that is definitely a problem. You'll see at the bottom, there's actually been an increase though in one study showing that they're able to access more behavioral treatments. And I really think that is because COVID, because the advent of Zoom, online access and so forth. And we're so much better able to reach people in areas where they really were these deserts where they didn't have access to resources. And it's been, so CHAD is one of the groups we've worked with a lot in the past, and they have really fantastic resources and support groups. And most of their meetings are online now. And so people can access those as well. Now you can see from this figure right here that if somebody has ADHD, there's a really good chance that they also have another disorder or challenge. And so as clinicians and teachers and parents, we really need to think about if we're looking at ADHD, we probably need to be assessing for other challenges as well. The younger group have the most with the behavioral disorders, so that's like the oppositional defiant disorder, and they may have conduct disorder. That's the 44% blue line. But anxiety disorders are also very, very common. So those also need to be assessed. And what we see as people develop, you tend to see more and more of this comorbidity. Learning disorders, we've always recognized as being high in ADHD. So that could be dyslexia, dyscalculia, dysgraphia. So keep that in mind, because that can also very much influence one's ability in the classroom. So this is from a study we just published within the past few weeks. This is from the CHARGE and the RECHARGE study. So Irva Hertz-Pachotto directed this and continues to direct it for many years, and I work on it in collaboration with Rebecca, as well as Debbie Bennett. And this was a study where we looked at kids who were brought in between 2 to 5 years of age, and we brought them back between 8 to 20 years of age. And we looked at whether or not they had ADHD, but not just ADHD, whether they had a particular subtype of ADHD. And there were 4 primary groups when they were originally brought into the study. On the left, On the left, you can see this is the autistic group, and then we have a developmentally disabled group. So these are kids with developmental disability without autism. So Down syndrome is one example. And then OEC is their other early concerns. And these are children who were initially brought in, and there was some thought that there was an autism diagnosis. But based on our in-depth evaluation, they did not meet criteria for autism. And then this group is really a general population group. And so what the blue bar shows is whether or not they didn't meet any criteria for any ADHD diagnosis when they were brought back when they were older. So the vast majority do not. But if they're in the autism or DD group or other early concerns, they have anywhere from a 3 to 4 times more likelihood of having it qualifying for an ADHD diagnosis. The red bars indicate if it's combined type of ADHD, so that's where they have the inattentive, hyperactive, and the impulsive symptoms. So we see the autism and the DD groups tend to have more of that. Where we see other early concerns is they have much more of the inattentive type, and same for the general population group. So perhaps these children, when they initially came in when they were younger, it may have looked like autism, but it may have really been ADHD symptoms. The hyperactive impulsive alone diagnosis is very rare. We typically see those in preschoolers in the general population, but we actually saw some children, a small group, who also have that in the autism and a little bit in the DD group. This is important because there are treatments being developed for subtypes of ADHD. So if we can get to the point where we can actually diagnose according to subtypes, we might have more powerful treatments to help these children. But for a long time, one could not diagnose ADHD within autism. That was not allowed by the DSM. It was really just the past several years we've been permitted to do that. And now we're trying to catch up by having improved diagnostic measures because we're still working on it. In some situations, it can be quite challenging to make that diagnosis. So shifting gears here, this is just, I put the slide in because I think it just really points to the importance of ADHD because sometimes people can minimize the impact of ADHD, but this was an alarming study that came out of the United Kingdom last year, and it's looking at adults who had been diagnosed with ADHD, and you can see for men, their lifespan is 6.78 years lower. For women, it's almost, it's about 8.5 years lower. So we don't necessarily think it's directly the ADHD, but it's the associated issues that they have, such as accidents. We see a higher rate of suicides, particularly in women with ADHD, and some other comorbid challenges. So to me, this is a really compelling slide. That means we really need to think seriously about ADHD, how we're screening for it, and also about making sure that we have treatments that reach people who need it. Now I'm going to shift gears again. I'm going to talk about some of our research. And we're really interested in our lab to do research that's very contextually relevant, that represents what sort of situations people experience in real life. And one of the projects we've been working on for several years now is how we diagnose distractibility specifically within ADHD, or just distractibility in general, because all of us, since the advent of the cell phone, are more distractible. If you're not more distractible and you're in the audience, you are rare. Most of us have a hard time. We get alerts constantly, whether it's mostly on our cell phone, but certainly even on our computers and so forth. And we just thought we really wanted to find better ways to measure distractibility within ADHD, but just also in our general population of kids. And we're particularly interested in the school setting because, again, this is where oftentimes we see the challenges. So on the left side, you can see here with the traditional ways of measuring distractibility error. This is a parent filling out a rating scale in a waiting room. This is oftentimes how it's done. And rating scales are valuable, there's no question about it, but there's also limitations. They can have recency effects in terms of the last experience they had with the child. They don't really break down contextually what's happening in terms of attention. The other way of measuring attention is using continuous performance tests or computer measures, and those can be highly precise, but they're very limited, and they don't really reflect the real world oftentimes. So like the example here, you press the button for go if you see a square versus if you see a round, if you see a circle, you would hold a response. And we felt like that doesn't really capture what happens to a child in a classroom. It doesn't really encapsulate what happens when somebody else is talking near them, when there's something else happening outside, a school bus going by, people talking outside the window, or mind wandering. So we wanted to see if we could improve upon that, and this is one from our most recent projects. And so this is built on years in our work with our virtual reality, and Patrick is here to attest. This has not been an easy project. We've had evolution over the years because they keep updating the software, but we've made some really good progress. And I only have 5 minutes left, so I'm just going to say really quickly, we're using this technology. It's helping us get much more refined measures. We're using eye tracking. We're using performance on academic tasks, and we're looking at what happens over time in a way that just cannot be captured by rating scales or computer tasks. And just real quickly, in terms of some of our other work, we've been doing a lot of work in fidgeting over the years. Hyperactivity has been is the most observable characteristic of ADHD, but many researchers haven't studied it. We long ago, and some of our colleagues saw that it seemed like when kids were moving more, they were actually doing better. So we tested that in the lab, and I'm showing this New York Times article because we got a ton of press from the New York Times, Wall Street Journal, Washington, all over the world we got press about this. But we are not responsible for the development of the fidget spinner. We've been criticized about that, but we looked at natural fidgeting with atrographs on the leg and the wrist. And what we show overall is that when there's more intense fidgeting, kids with ADHD are actually seeming to be doing better. We followed this up with adults with ADHD. Again, we saw fidgeting was associated with better performance. And the take-home message for me is that hyperactivity is not a bad thing. It used to be, I would see parents all the time trying to tell their parents to sit still and be quiet, and I can understand in certain contexts that's important, but also people have to understand it's not under their control, the hyperactivity, and find ways to have them naturally express that. So exercise, there's lots of good evidence for exercise. We've been looking at fidget tools. The data are suggesting that it might not always be as helpful as we'd like. It's really that sort of intrinsic movement that we think is probably most helpful. And we're trying to explore that right now in our lab. This is a really exciting clinical trial I'm involved in. It's funded by the SBIR grant through NIMH with my colleagues Jeff Nyquist and Duye Tadden. And Jeff Nyquist owns a company called Neurotrainer. And it's really cool because what it does, it integrates cognitive training with physical movement. So kids are using a virtual reality headset, they're doing it at their school, and they're acting out their attention training. And so they combines the best of both worlds. We've tested over 100 children. It's taking place in 3 schools in the Midwest, and we're crunching the data right now, and we'll see what happens. But this has the potential to reach a lot of people. Because it's already commercially available, and a lot of school athletes, not just school athletes, a lot of the pro teams, the baseball teams have been using it for years. So it's a cool kind of intervention that kids like. So we'll see what the data show though. This is a little bit, our workshop this afternoon is about how we've modified our the HOPS training group for addressing and improving executive skill functioning in ADHD. This is part of a meta-analysis that our team was part of, looking at parent training, which is, as I said, very common in terms of interventions. And the take-home message for this is that it's really focusing on reducing negativity and building affection. If you have to really focus on a small area, that seemed to be that made the difference in terms of improving functioning for these kids. If you can improve positive behavior in parenting, that should naturally reduce the negative. But if you're seeing parents who are doing a lot of yelling and inconsistency in how they apply their interventions and their rules with their children, that's an area to focus on. And it was most strongest in those families who have the most non-constructive parenting. And these are our resources that we have, and I think they are in the Padlet. And this is just some of the many amazing collaborators that I have and team members. Many of them have gone on, many of our trainees, but it really does take a village, and I'm really grateful to have such an incredible team that make me look good by producing this kind of data to present to you. And thank you for having me.
The first question would be, would you recommend Fragile X syndrome testing for a child with Down syndrome?
So we have seen many individuals with Down syndrome and Fragile X syndrome together. I think a key thing is whether the person with Down syndrome has autism. And all of the ones with Down syndrome and Fragile X together have had autism. So if there's a positive family history, particularly with premutation disorders, and the child with Down syndrome has autism too and may be more affected, then I would test for Fragile X.
Also, there is a question specific to advances in CRISPR, CRISPR technology. How do these advances impact Fragile X carriers or infants?
So CRISPR directly impacts the DNA, most types of CRISPR. The problem is sometimes the CRISPR cutting can be a little inaccurate, and then you can't reverse it if there's a problem. So they are developing some CRISPR techniques that cut and splice the RNA. I think that this technology can be good. It's been studied with an AAV virus having a modified CRISPR gene that infects the CNS. So there are techniques that work in animals, and eventually this will be available for patients. It can also be helpful for premutation disorders. My husband's working on a transplicing where you develop a message that has a much shorter normal size CGG repeat, and you get it into the central nervous system, and this transplicing can take over the RNA that has too many CGG repeats on it, and then it can take over and then produce just a normal amount of messenger RNA. So there are some really exciting new RNA techniques, but also CRISPR techniques, CRISPR-Cas9 techniques. So you'll see much more of this in the future. I think the FMRP TAT that I mentioned that can be injected, I think that'll be coming out first, probably within the next 2 years.
Okay, great. A follow-up to that, just in general, do you feel CRISPR is an ethical treatment?
Yes, I do, for the families that want to utilize it, absolutely.
And then one final question for you. If there is a parent of a child with Down syndrome and autism, do you recommend that the mom receive testing for Fragile X?
Yes, the mom has to be a carrier for the child with Down syndrome to have a full mutation. However, we have seen some individuals who just have Down syndrome in the pre-mutation, and in girls that could come from a carrier father too. So if, one could test the mother first to see if she has a pre or a full mutation herself.
Doctors say that they cannot test for ADHD until the child is 5 years old. Is there a particular specialist that can test before 5 years old?
It's the same standards what we use right now. Is we use the rating scales, and it's always, we're always saying what we use is multi-informant. So if they're in kindergarten and so forth, to try to get a kindergarten teacher as well. And behavioral observation can make a huge difference. It's just that if you're observing them in the clinic, it may not necessarily represent what they're like at home. So that's why you can try to, but there's no specific computer test or blood test or anything like that. It's a really solid interview, and oftentimes with ADHD, what I'm doing diagnostically is ruling out other reasons why we might be seeing problems. And at that age, you're probably seeing mainly hyperactivity, because it's hard to assess the inattention and some impulsivity at that stage. So are there any other factors that might be causing it, particularly if it's a sudden onset? Because we should have seen it for several months, if not a year, and so forth.
Are there parenting books that you recommend for young parents with children that show early signs or have any of the issues that you spoke about? So maybe a couple of titles if you have.
So parenting books, is it for just general understanding, or is it for actually how to do some interventions and so forth?
I think either.
Either,
Great.
okay.
Yeah.
Well, there are, I don't know if you consider them books, but there is the P Positive Parenting Approach that's online. There's the Incredible Years that are both really well done. And those are classes. They have a variety of different ways that they deliver the materials. Also, the state of California, which I did not include in my slides, has released for free a lot of parenting resources. So we should definitely check into those. Those are, I think that was the Commission on Behavioral Health. So maybe by the end of the day we can find those, but there are tremendous resources. Some of them are videos, but some of them are actually where you can connect one-on-one to speak to an individual.
Okay, we'll follow up and add those to the Padlet. Rebecca, do you have an answer?
Yeah, I was going to say, I don't know of any books, but I would say the same thing in terms of resources. There's a lot out there, apparently in California and elsewhere, on the first 1,000 days, which does seem to be a really important period where you need to think a lot about how to make the best opportunities and for your child. And so I think those would be a good thing to look into as a parent. But of course, it doesn't end there after that first 1,000 days. And so there's, it continues on. And then at that point, there's a lot of other separate resources that can inform that pathway, I guess.
And one I forgot was understood.org. It's very visually compelling, and it really covers over a broad range of challenges. So if people look at understood.org, it's a great organization.
Randi, do you have any recommendations?
No, I agree with everything they say.
And we
Good
have
answer.
some resources of the Mind Institute.
Yes.
So we have our own resources too.
Yeah,
We have
resource
videos. Patrick helped us curate some great videos that are understanding ADHD and so forth. So we have in many different areas.
For sure, yeah, do check out our website.
Also, we link directly to the ADHD and Fragile X specific resources that you can find through the Mind Institute website, but there's a link on your Padlet as well if you want easy access to get to those. I'm a transitionist. Dr. Schmidt, I have a question for you. What do we know about the impact on stressful experiences in gestation on neurodevelopment?
Yeah, that's a great question. So it's also a big area of focus for all of our studies. So yes, any kind of stressor can have an impact on maternal health, and in turn, if she's pregnant, it can have an impact on their developing child in a lot of different ways. There's kind of inconsistencies across a lot of studies in terms of what timing is the most relevant and maybe what type of stressor is the most important. But it does look, at least in our MARBLE study, we looked at a few different markers of stress in the mother, including measured cortisol levels in her urine, but also some, the one that came out, and just stressful life events that happened during pregnancy that she reported on prospectively. And then also just her own perceived stress scale ratings, so how stressed she perceived she was at various times during pregnancy and afterwards. And that was the one that was the biggest marker. So it seems like it might not just be like, everyone reacts to a stressful event differently, and I think that's why the literature is a little mixed on like in terms of a stressful life event, how it impacts both the mom and the baby, because everyone reacts a bit differently. And so it's really how the mother perceives her own stressful situation and how stressed she feels as a result of it that seem to be the most associated with the child's later neurodevelopmental outcomes. And that was also something we were very cognizant of for the wildfire events, because a lot of them, again, lost their homes. And so not only are they dealing with, smoke in the air, but there's, huge life changes if they had to move, if they lost their neighborhood and all of their contacts, and or they could have lost a pet or a family member. So we did also collect information on all of that. We haven't looked into it too much. We also have the perceived stress scales for those moms, and we do see that they were stressed at the time of the wildfire events. So, and that was the one thing that kind of persisted long term, like up to a year later, she was still reporting high levels of stress and anxiety. So that's something we want to look into more. But I think the bottom line is that, stress isn't good for anybody, including developing children and their mothers. But then it's also, you don't want to tell them, don't stress out about being stressed, because it's hard to reduce stress on a lot of cases. But there are, a lot of thoughts about how to maybe stress-reducing behaviors and things that can be done to kind of have good support systems and things like that. That might be more important to help reduce the consequences of being stressed.
So women that get diagnosed with ADHD later in life, are they often misdiagnosed due to symptoms of perimenopause?
Oh, that's a great question. And there is actually a lot of research right now, actually in the Netherlands going on in that area. So it's not that clear right now, but it's a very active area of study. And this is when you look at the trajectory, and you really, because ADHD symptoms should have arisen before the age of 12, although sometimes if you're really bright and have skills in other areas, people may have compensated. So sometimes that's why they're not emerging until really in their young adult or even middle adulthood. But then you do get that issue, and Randi maybe could speak better about this, about the hormonally, or maybe Rebecca, you could, is that there's also, there are changes physiologically that occur that mimic some of the same challenges associated with ADHD. So hormones are related to memory and attention and so forth. So you get this cascade, and I think that's where it really gets hard to differentiate. But some of the tools may be the same, but there's actually a group where they're collaborating with gynecologists to help get the referrals to their clinic to study some of this. So it's a fascinating area, and it's certainly something where we need some clarity, because it has very different implications in terms of interventions.
What are first steps if you believe you or your family might have Fragile X?
So the first steps is you would discuss this with your doctor. The problem is a lot of doctors don't know that much about, especially premutation disorders, and they mix up Fragile X syndrome and premutation often. And so it's helpful for families to educate themselves, like going on the National Fragile X Foundation website, fragilex.org, and you can explain to your doctor what you know and then ask for a Fragile X DNA test, and then usually they will order that Fragile X DNA test. And for a child who's not speaking by age 3 or has very severe hyperactivity, in my clinic I have wildly hyperactive kids under 5, wildly, that are tearing apart my office. You hardly need a checklist to know that they're wildly hyperactive and out of control. And so I don't like to use stimulants before age 5, but oftentimes guanfacine or clonidine has an overall calming effect. And then related to stress, a lot of the premutation moms have a lot of stress, and the most stress comes when they are experiencing aggression from their child. So if their child is hitting them, or they're getting physically injured by either the child or the husband or someone else in the family, that you really have to focus on and bring treatment to the mom and interventions for the child so that everybody doesn't get stressed out. And a lot of our female carriers have ADHD later on in life. It's actually there in childhood, but they're able to compensate pretty well. But sometimes when they go through menopause or have a big area of stress, and menopause for many of these women has significant cognitive and behavioral problems associated with it. So these kinds of traumas or physical changes really bring out some of these symptoms. But it's easy to do a Fragile X DNA test anytime you're concerned about it. You just need a doctor to order it, and if one doctor doesn't do it, you can go to other doctors or come to me.
The UC Davis Mind Institute was founded in 1998 with a promise to reduce and prevent the disabilities that can be associated with autism and other neurodevelopmental conditions. Every day, our clinicians and researchers make progress on that promise. Our groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other conditions associated with disability are helping affected individuals achieve their fullest potential. Please visit our website or our social media platforms to find out more about current studies, upcoming events, and how you can help make a difference.